Okay, why don't we get started? Happy Valentine's Day. You gave me your Valentine's Night on Tuesday. I was going to give you a Carnival Cruise trip, but I got stuck. I don't think anybody wants that one. Okay. All right, so we're going to start a new topic today, particularly if we can see all of it. Let's see. This is a topic you should be already familiar with quite a bit from introductory biology. You should know the basics of the phospholipids, what goes into the membranes. If you don't remember that stuff, then I posted a background PowerPoint and a background outline for you to refer to if you go, what is a phosphatidylnyl anything? What does that mean? You need to, pardon me? Oh, okay. You need to know that stuff, but this is not a biochemistry class. You don't have to memorize every single head group. In other words, if we're talking about phosphatidylcholine, you're not going to have to recognize the structure of choline, but you need to recognize the structure if I say phosphatidyl, because half the phospholipids are in that particular structure. So you can just go get the PowerPoint and look it up if you've forgotten a particular thing. Or if you've never had it, maybe you didn't take it from Dr. Dini or whatever, you can have a little brief refresher by going over that. Okay? So you'll see as we go through here what components seem to be important and which are less important. But memorization is not something that we're really into, except for the poster children, as you well know. If you want to go memorize stuff, then go to biochemistry. Okay, so here's what we want to do. Again, you decide how much of this review you need to do, and I'll be happy to talk with you about the organization of membranes. I love it, actually, but we don't need to take up class time by rehashing something you should have had in high school. Okay, classify membrane proteins in appropriate categories. That's something we're going to focus on because now we're into protein function. Membranes, of course, is going to construct half of the rest of this course because they control what goes in and out of cells, what goes in and out of the nucleus, trafficking. There are surfaces for enzymatic reactions. You can't move a protein to certain places unless you put it in a membrane and move it. So it's a very dynamic topic that we need to have a very basic vocabulary. And we also need to have a protein toolkit, things that we can do to analyze proteins that are in membranes. And so we'll look at that. Although this is not, obviously you know this is not a methods course, we do need to have some of that vocabulary to go forward. Okay, so this is the lipid mosaic, the fluid mosaic model. Okay, the essence of this is that there are two leaflets, okay, that construct a bilayer. That's why it's called a bilayer. The names of those leaflets you should know. The one that faces the cytoplasm. So this is an electron micrograph, okay? So that means this one down here is facing the cytoplasm. So guess what? We call it the cytoplasmic leaflet. What do you call the other one? Anybody know? Your book calls it the non-cytoplasmic leaflet, which is ridiculous, actually. It has a very good name. It's called exoplasmic leaflet. Okay? So any membrane-bound structure, including the ER, the Golgi, mitochondria is actually a little tricky. Let's do that one in a minute. And let's go ahead and finish what this fluid mosaic model really entails. So it's got two leaflets to make a bilayer. It's extremely fluid, hence the term fluid. The fluidity is determined by the types of lipids that are in the membrane. Okay? So this is moving around. In fact, this cannot function if it freezes. If it goes from a sol state, which is the soluble liquid state, into a gel, things die. Okay, so you don't want it to become a gel, so you need to keep things fluid. And we can actually measure that fluidity with various techniques. It's a mosaic of proteins. The proteins are the mosaic part. They're little chunks, little icebergs floating around in there. And we now know that it's not limited just to chunks of icebergs. There are lipid rafts that are semi-ordered liquid state. It's called liquid order. So we now we can describe the fluidity of the lipids in three different phases. Sol means it's completely fluid. Liquid ordered means it's in a raft, a liquid raft. And then gel means it's frozen or non-functional. Okay. All right, so again, just go back to the naming stuff. Obviously, something like, let's draw a cell. This, okay. There we go. All right, and so you have the ER, which is a lot of little sacs. And so you already know that this is the exoplasmic compartment, right? And we found out that that's where disulfide bridges form, right? So exoplasmic has certain characteristics. It's an oxidizing environment. This is the exoplasmic environment outside of cells. It's the extracellular environment, okay? So the exoplasmic leaflet always faces that compartment, okay? Now that's pretty simple. So if we blew this up, took a little chunk like that, and we'll push it up here, make it a little bit bigger. It's like this. And so you got a bunch of little lollipops. Those are the lipids. Okay, and then two little sticks coming out. Okay, so this one facing the cytoplasm is clearly the cytoplasmic leaflet. This one is the exoplasmic leaflet facing this direction. What about a mitochondria? Let's do that. So this is usually one that gets people. So the outer membrane is pretty unfolded, but the inner membrane is folded into cristae invaginations like this. Okay, so that's the outer membrane. This is the inner. And then, of course, this is the intermembrane space, right? All right, so this is a leaflet. And so what do you call this bilayer that faces this direction? Well, is it facing the cytoplasm? Absolutely. So the outer mitochondrial membrane is easy. Okay, so this is the cytoplasm. So this is a lipid bilayer. This is the outer. This is the cytoplasmic leaflet. Okay, the leaflet that faces the intermembrane space is the exoplasmic leaflet. So the intermembrane space of a mitochondria is exoplasmic compartment, even though the pH and small ion concentration is exactly the same as the cytoplasm. That's confusing to people. Okay? Now let's do the inner membrane. Remember what this is. This is an ancient bacteria that we enslaved a long time ago. And so the cytoplasm of the bacteria would be where? Where are the ribosomes in a bacteria? Right here. That's the matrix. The matrix is the cytoplasm of the bacterial thing that we enslaved billions of years ago. Okay? So that actually makes it easy. But so this leaflet, we can draw a leaflet now, right? The leaflet that faces this direction is cytoplasmic. The leaflet that faces the intermembrane space is still the exoplasmic leaflet. So that's the most difficult one. You might think about doing the nuclear envelope with this exercise and figuring that one out, but it's not too complicated. So are there any questions about this vocabulary? I want to do the tricky ones, and the other ones are just straightforward. Okay, good. All right. So there's three important properties that this structure yields. Let's get a different viewpoint here. So those are the standard phospholipids, and so you're going to go back and review that structure if you're unfamiliar with it. But since a lot of the phospholipids are constructed of this phosphatidylinositol or phosphatidyl head group. So this is phosphatidylcholine because the head group is choline. There's a phosphate diester bridge. There is a glycerol molecule is the backbone. So all of this is polar. If it has a hydroxyl group, it's got to be polar. Phosphate is charged negatively, so it's definitely polar. And the head group is polar. Okay. This is the nonpolar part. Okay. So these are the two fatty acid tails. If it has this structure, it's a phosphatidyl something. Okay. And there's different kinds of phospholipids that we'll get into. So this is the picture that I wanted to see, to show you. And so the properties that this bilayer has is the middle here is extremely hydrophobic. It's extremely disordered, and we want it to be disordered up to a point. That contributes to the fluidity. So all those fatty acid tails are just mingling around that are very disordered. That's going to keep water and ions from diffusing across this structure. So it's, number one, the characteristic of this lipid bilayer, it is highly impermeable to water and ions and other polar things. Number two, it's got a really high electrical resistance. Ion and electricity in living things is carried by ions, and so that means ions can't cross this unless you put a protein bridge there known as an ion channel. So you got to have proteins to give electrical conductance. So this is very highly resistant. Okay? The last thing is, if you poke your little finger in there, you take your little molecular finger and poke a hole in it, what happens? Do you die? Does the cell die? No, it reseals. It's self-healing. Okay, so it reseals. All right? So those are the three important properties that structure creates. The other thing is, if you look at this, the polar head groups are these red things. Choline, phosphate, the backbone, the hydroxyl group of cholesterol is a single head group. Okay? This is very spiky. It has a surface to it. And what do we do with surfaces? They're binding sites. And so the water gets down in here and intercalates. So this is a really manageable surface that a protein can recognize. So there's meaning to this order of this little spikiness. Okay, so it's a binding surface. We can do things with it. And we will do that when I'll show you how peripheral proteins bind to membranes. Okay, this is the other thing I've already mentioned, is that we do have three states. This is a lipid ordered area, and we call those lipid rafts. The lipid raft generally is comprised of both the exoplasmic and cytoplasmic layers, and it's obviously it is going to be less fluid than that stuff over there. So these proteins are going to be moving around faster than these proteins. And what it does actually is it will sort certain types of proteins into this lipid raft. There's going to be people that jumped into the ocean and they hopped onto this lipid raft, particularly GPI anchored proteins that we'll talk about. If you don't know what that is, we'll talk about it. It's a lipid anchored protein, tend to associate in lipid rafts. Signaling molecules like the insulin receptor and certain types of receptors tend to coalesce. So it's a way for proteins to assemble into a complex that's going to have a function. And so we will look at that, particularly when we get into signal transduction. So this is not useless. And this whole raft is moving around, even though it's slower than all this stuff over here. Okay, questions about raft structure? It's also shown this way. This is actually, if you see this type of structure, it's not a great way to look at it. This is from a different textbook, see, not in your book, okay? And so what's wrong with this picture? The lipid raft is only on one leaflet. What happens is when these lipid rafts form is that this is highly enriched in two things, cholesterol and sphingolipids. You go, I don't remember what a sphingolipid is. Go back to the background PowerPoint and look at it. They tend to be very stiff and very long. And the length of the fatty part of that sphingolipid causes it to interdigitate with the cytoplasmic leaflet. So the two leaflets are now coupled as a raft, moving together. So things can go, for proteins as you saw, and this is a good, even though it's a simpler diagram, it's actually more accurate. A protein can span this entire raft as a passenger on the raft. Okay. All right, good. So we know it's got special properties that make it less fluid than this stuff over here. One other thing about the heterogeneity of this is a good thing to know is the phosphatidylserine is enriched in the cytoplasmic leaflet. So this is very asymmetric. The exoplasmic leaflet and the cytoplasmic leaflet are very asymmetric. All of the phospholipids that contain glucose or sugars are always in the exoplasmic leaflet. That's another good asymmetry to remember. And all of those are discussed in the background material. Okay, this is what we really want to get to today. This is the main topic. How do we categorize all these players that we're going to run into? We will use this vocabulary for the rest of the class. the rest of the course, so it's good to understand it well as we go along. And you should have already had this, so we're going to go through it, but I want to give you my interpretation. That might be a little different than the focus that Dr. Deeney had. Okay, so the first type, this is a single pass transmembrane protein. Okay. Now, broadly, we're going to divide membrane proteins into two big categories. So if you can see a concept map forming in your head, those are the first two divisions, integral and peripheral. Okay. So let's talk about the integral membrane proteins first. First of all, what makes a protein integral? It's the ability to remain in the membrane under conditions of high salt, high pH, chelators, things that would naturally break up the hydrogen bonds. The peripheral proteins will dissociate. The integral proteins will not. They stay in the membrane. To get an integral protein to leave the membrane, you have to use detergents. You have to use soap that disrupts the membrane and releases the protein. So integral membranes are integral parts of the membrane. They have lots of hydrophobic residues. See this alpha helix? You can guess what those R groups are projecting off of that alpha helix. Let's take a look at it. So here's one of the things. One way to get a protein to go across the membrane is to use an alpha helix. Okay, I think I have a, yeah. So the poster child is usually glycophorin. This protein actually exists in red blood cells as a homodimer, but we're not showing that here. Okay, so this is the alpha helix. It's about 21 to 23 amino acids, and that's a very important number because that's the number of amino acids giving the how many residues it takes to go around and around to get this across this lipid bilayer. So it takes about anywhere, the span is 19 to 23 amino acids. That's usually what we look for the number of amino acids in this alpha helix. And the way you're going to deal with the R groups is they project out radially, correct? And so as long as you're in this lipid area, you're going to want to choose from family VWR, GP, that whole hydrophobic group. And all you have to do is stick them projecting out of here radially as you penetrate this membrane. The minute you pop out, then you're going to start using polar and charged amino acids, polar and charged. Okay. So the characteristics of these are very polar hydrophilic residues. These are hydrophobic, and these are hydrophilic again. Notice something else. The sugars are always on the exoplasmic domain of a single pass membrane protein. Sugars, disulfide bridges, are always on the outside. They help protect that protein from the onslaught of proteases and mean things that are gobbling on the protein. They keep it tough. Okay? Why would you have sulfhydryl groups here if you can't form disulfide bridges? Okay, hold that thought. We're going to use them a little bit later, okay, to make other membrane proteins. Okay, so we have single pass and then we have multi-pass transmembrane proteins. Again, we're using the same secondary structure to get across the membrane, except this time we're going to go in and out and in and out and in and out. Okay. This is bacterial rhodopsin. It's unfortunate that your book uses a bacterial protein. In us, a good example of this has 7 transmembrane alpha helices would be a G protein coupled receptor that we will talk about. G proteins are those things that like you saw for eukaryotic initiation factor 2 that are controlled by GTP binding and GDP binding. And so we will talk about G protein coupled receptors a lot. But they all have the same, I would call it a motif. It's a 7 helix motif. Okay. So bacterial rhodopsin is our multi-pass membrane protein example, but there are many others. Okay, the second way to get a protein across the plasma membrane and do it professionally is to use a beta barrel. This is a motif that you know about. This is the porin. that's in the outer mitochondrial membrane. You now know that this is, if this is the extracellular, well, they're doing bacterial proteins. Okay, so extracellular space, periplasmic. But let's pretend that this is actually in the outer mitochondrial membrane. And so actually, this is the cytoplasmic side of that outer mitochondrial membrane, and that would be the exoplasmic side facing the intermembrane space. Go on and practice that at home, drawing it to you. Convince yourself how you would situate a porin in a eukaryotic mitochondria. And this allows things to move through here based on size exclusion. If it's, I think it's about 5,000 molecular weight or lower, it gets to go through for free. There's no transport. It's diffusion only. So it's not very selective except by size. But what that means is this is the cytoplasm, and that's the intermembrane space. It means the pH in the cytoplasm, pH 7, is the same as the pH in the intermembrane space of the mitochondria. It's the same sodium concentration, chloride, blah, blah, blah. All the ions are just moving freely through this thing. But how do you construct that? So you're taking these are called the staves, but you also would call them what? If this is a beta sheet, what are these called? These are the strands. It's a multi-strand beta sheet that's rolled up into a beta barrel. So let's take this edge right here. So this one's running right along this edge, and you know that the R groups are going to, if they project into the lipid, they're going to have what property? It have to be family BWR, GP, and C, or whatever it was. Okay, so they have to be hydrophobic, but remember they go in and then out. They alternate on each side of the strand, correct? So every other one is good. So this one's going to be hydrophobic. The one that's facing the lumen of this thing is hydrophilic. So to identify the strand that is participating in a porin, you're going to look for alternating hydrophobic, hydrophilic, hydrophobic, hydrophilic. And that's how this gets across. Okay, so it's a really different way of going across. And you don't need, how many amino acids do you think it takes to get a strand to go across here? Let's just one that's right on the edge here. Let's say this, more or less than an alpha helix? Less, because it's not rolled up into a helical. It's just straight. It's a little bumpy, right? It's going up and down, but it's short, much shorter. Okay. So the porins are our poster child for the beta barrel motif that goes across the membrane. Okay, porins. So we've done this one. We've done this one. So this would be glycoform, bacterial rhodopsin, or G protein coupled receptors. This is porin. Now we have this guy. Okay. There's not too many of these. This is embedded. It's called an integral membrane protein, but it's embedded. And you can see how this is going to work. You're actually going to take the alpha helix and turn it sideways. Instead of being up and down, it's going to kneel into this cytoplasmic leaflet only. So that's the characteristic of this integral motif, is that it only goes into one or the other leaflets. And typically, all the examples I can think of are intracellular proteins, so that thing is going to kneel into exclusively the cytoplasmic leaflet. And how is that going to work? As you go around the horn, you go round and round. If you're projecting an R group that goes into the membrane, you have to pick one of those hydrophobic residues. But what about these that are facing the cytoplasm? You get to pick a hydrophilic residue. So depending where you are going around and around that corkscrew, that's how you choose the residues that project into the cytosol versus those that project into the cytoplasmic leaflet. Okay. Just to give you some more details, I don't like to give people proteins and not talk about them. This is the protein that synthesizes prostaglandins, and prostaglandins. There's two classes of prostaglandins. There's the good ones, and then there's the bad ones. Okay, so the good ones are the ones that protect your stomach lining from acid. Okay, so what the prostaglandins is signaling, it's like a hormone or a cytokine. When it's released, what it does is it tells the mucus cells in your stomach to release mucus that coats it prevents your stomach from being eaten by the acid that's being produced. So if you inhibit that enzyme, what happens to the mucus in your stomach later? It decreases or is not produced. And then you get things like ulcers happening to you. That's exactly what happens when you take NSAIDs, non-steroidal anti-inflammatories. So like aspirin, ibuprofen, they're inhibitors of that enzyme. Non-selective inhibitors. They inhibit the good and the bad and the ugly. Okay? So one of the problems with aspirin in all NSAIDs is that they are non-selective inhibitors of prostaglandin H2 synthase. Okay? What's another good thing? Platelet aggregation. I mean, you need to be able to clot your platelets. Okay? But you can go overboard and have clots where you don't want them and give people strokes and heart attacks. Okay? And so that's why low doses of aspirin are good preventers of strokes in that clotting process. Just a little bit of aspirin inhibits the platelet aggregation just enough to lower your probability of forming stroke components. Okay? So platelet aggregation is a good property, but not too good. We don't want too much of it. What else can we do with prostaglandins? Ovulation in the menstrual cycle, okay, is the way that the ovum breaks through the wall of the ovary and goes into the fallopian tube is the prostaglandins are released that degrades the tissue that allows it to go through into the fallopian tube. So that's a good prostaglandin. So obviously if you're on high levels of aspirin therapy, are you going to be able to ovulate? Probably not as well. Okay. One of the major contractions producers in the uterus during birth is prostaglandins. So that's a very useful thing. So we need to make drugs that are a little bit more selective. What are the bad things that prostaglandins give you? Inflammation. So the reason you typically get a headache is because you're releasing inflammatory cytokines, causing pressure on blood vessels, makes them leaky, causes all kinds of problems. You take aspirin, right? I mean, and you can take fairly high doses. Also fevers. Aspirin's a good fever reducer. People that have arthritis. But what's the downside of taking that aspirin? It's a non-selective NSAID, right? But if we know a little bit about the biochemistry of that protein, which includes a core enzymatic component called cyclooxygenase. Cyclooxygenase, I'm not sure how to spell this, maybe it's just one O. That's a component, that's one of the key components in prostaglandin synthase. And what happens is there's two types of enzymes. There's COX-1, and then there's COX-2. COX-1 is the good one, okay? That's the one that's involved in ovulation, birthing, platelet aggregation, and producing a nice mucus layer in your stomach. That's the good one, okay? And it has a particular, where's the rest of my kits? Okay, we don't have it. All right, so we can draw it's like this. It's a protein, and it has a very narrow binding pocket right there. Okay, now, the bad one, this is the bad one. This is the inflammation one. Causes fever, inflammation, arthritis, whatever. Okay, its difference, it's the same basic molecule, but it's got a wider binding pocket. Okay. The problem is, if you take aspirin will fit into the good place, and it fits into the bad. So it inhibits both of them equally. And that's not good. So what they developed are COX-2 inhibitors. Okay? And they give them to people that have really painful joints, arthritis. It's a wonderful tool because what they did is they made it fat. That inhibitor will fit into the binding pocket only of COX-2, but notice it won't go into the slender binding pocket of COX-1, and it will not touch your platelet aggregation. It will not mess up your stomach lining, you can ovulate, you can have birth, whatever you need to do, it's good. So that's an amazing way that chemistry helps inform us in making drugs. Okay. But back up to the story, prostaglandin H2 synthase is a good example of an embedded integral membrane protein. Okay, questions about that?
What did you say it increases the degradation of to help ovulation?
Oh, it's the wall of the ovary
Oh,
that's going, yeah.
Why do they still sell the COX-1 inhibitors like aspirin, and why not just the COX-2?
Because if you take a little bit of aspirin, it helps reduce inflammation, but also you want that platelet aggregation to not be quite as strong. So a little bit of inhibition. And you get a bonus is that it's going to help decrease whatever local inflammatory things. So a little bit, and it's cheap. It's really, as you know, enteric-coated, so you can take enteric-coated aspirin. That's why you choose that when you take that aspirin to prevent strokes, is it'll get through your stomach without chewing on your stomach. Yeah.
Can you build up a tolerance?
Pardon me?
Can you build up a tolerance to aspirin?
I don't think so. I think, you know, there are people that can make an antibody against anything. Some of us are really good about making antibodies to things we shouldn't make. So I would not say exclusively, but I'd say it's, for the most of the population, it's just not antigenic. Okay. Good. Where does that bring us? Okay, so that we're still in the integral membrane protein. And now we're going to go back to an old friend of ours. Finally, you get to see where C-Sark lives. Okay. Now this is the lipid bilayer. The category of integral membrane proteins is the lipid anchor group, and this is a pretty large group. So let's go through this. This is C-Sark, and C-Sark is a cytoplasmic signaling protein. So this must be the cytoplasmic compartment. In fact, most lipid anchored proteins are in fact cytoplasmic, with a couple of exceptions that I need to point out. So if the protein is going to face the cytoplasm, then that means this lipid anchor is going into the cytoplasmic leaflet only. It's only long enough to go across just the first one. And there's a couple of different lipid anchors that are used. What SARC does is, it actually, all of these that I'm going to show up here, there's two anchors, two lipid anchors, even though this cartoon does not show it. So one type of anchor is a myristic acid. That's a fatty acid tail that's got, I don't expect you to know the number of carbons, but it has 14. Okay, and so it's got 14 carbons, and then it's linked to a glycine residue, which is kind of weird, but that's how it attaches to the protein. And that just sticks into the membrane. Is it embedded? No, it's going in vertically. It's going to shoot. It's almost like a little fatty acid tail that just lost the rest of the phospholipid. Okay, but that's good enough to hold on to SARC. And what's SARC? It's a protein kinase that phosphorylates. It's a tyrosine protein kinase. It phosphorylates tyrosine residues on other proteins. But it does that to start a signaling pattern. Okay, so it has to face the cytoplasm, because all of its binding partners are in the cytoplasm. Okay, it uses a second anchor, as do both of these proteins, and that second anchor is interestingly attached to a sulfhydryl group. So this is another usage of cysteines that do not involve disulfide bridges. Well, that sounds like a test question if I've ever heard one, right? Give me some examples of what cytosines are involved in protein structure. And naive people that are in biochemistry, no, I'm kidding, they'll say, it's a disulfide bridge. And you go, well, what about the rest of them? You go, well, there are no other reasons. Well, there are. Lipid anchors is a good way to anchor that protein, which is SARC. The second lipid anchor is connected to cysteine residue. And then this is a palmitic acid anchor, palmitoyl, okay, palmitoylated. And so this has got, I think, 16. This one's 14. That's the only difference between myristic acid and a palmitic acid. It's a little bit longer. But it takes two of them to really hold that protein into the membrane because it's not going across. It doesn't have a good grip on the membrane, does it? It's only got one. It's just like the embedded protein. It's only in one lipid, one leaflet is the word I'm looking for. Okay, so we know what this does. We know how it's anchored. Now let's talk about RAS. And RAS is another signaling molecule that's really important in cancer biology because this is part of a signaling pathway that tells cells to divide. So this is CRAS, this is cellular RAS. Okay, and there are viral components of both of these that will give a person cancer if they're exposed to it. Okay, so you can shorten CRAS just down to RAS. If you're talking about SARC, you don't say C-SARC, you just say SARC, RAS, whatever. So this is a protein that's going to do something. It's actually a trigger to a different pathway that we'll talk about. But it's a signaling protein. And how is it attached? Well, it's got two anchors. It's got the palmitic anchor that it shares with C-SARC. Okay? And then it's got this is what I call a rubber tail because it's made out of farnesyl units. These are isoprene units that you're supposed to know if you took organic chemistry, but you probably forgot. But you make rubber by putting isoprene units together. Rubber, as you can imagine, is exquisitely hydrophobic, and so it goes into this membrane like a piece of butter. It loves that membrane. But I like to distinguish it and say it's got a rubber tail. So RAS has a rubber tail, SARC does not. But they do share this type of palmitic anchor. Okay? How do you attach it? You attach it again. That's another use for a cysteine, is another lipid anchor. Now here's a tricky one. What non-anchor, lipid anchor function that has nothing to do with disulfide bridges have we left out if we're informing this biochemist? Who probably knows it, but we just got through talking about it. We talked about gene regulation. What else can you use a cysteine to hold?
Zinc.
Yes, zinc finger. It has nothing to do with the disulfide bridge. It has nothing to do with the lipid anchor. It holds that protein into a motif that binds to DNA and dimerizes. So that's really good. All right. Now I can't ask that question, though. Come up with something else, I guess. Okay, before we go on, let's take a break because we're about to go into... actually, I'm going to give you a few more lipid anchored ones because I didn't finish them. And so let's take a break first. Okay, why don't we get started again? I forgot to actually cover this last example of a lipid anchor proteins, and these are important medically and biochemically and physiologically. This is the GPI anchored lipid anchors. It's glycosyl phosphatidylinositol anchored protein. That's why it's a GPI, right? So what it is basically a traditional phosphatidyl, group. That means it has two fatty acids, and then it's got a classic glycerol backbone connected to a phosphate, and then the first little sugar residue up here, it's actually a sugar alcohol, is inositol. And this is bizarre because most phosphatidylinositols are exclusively in the cytoplasmic leaflet. This is in the exoplasmic leaflet. All the GPI anchored proteins are in the exoplasmic leaflet. Okay? And then you don't need to know that detail of structure. Basically, it's a phosphatidyl phospholipid that has a string of sugars, and it's got a phosphatidyl ethanolamine that then connects it to the protein. So the protein structure is like a little kid with his two legs standing in the lipid waving a balloon. Okay, his arm is all that jazz, that sugar chain, and then the balloon is actually just waving around is the protein, coffee, the protein. Okay, so what's a good example of this type of protein? Well, a prion is a good example of this protein. Prions are naturally found in the nervous system. They're membrane proteins that face the cerebral spinal fluid, but what's happening is these prions can come into contact with abnormal prions, scraping prions, and that gives a person Creutzfeldt-Jakob syndrome. mad cow disease to you. Okay, so if it's in an animal, we call it mad cow disease. If it's in a human, it's Creutzfeldt-Jakob syndrome. And what happens is the bad prions interact with the good prions and they misfold and causes neuronal death. So these people basically lose their mind. Okay, so mad cow disease is caused by the prion. Another good example is alkaline phosphatase that's in your intestine. Okay, so you can make this protein do anything you want. So in this case, it's alkaline phosphatase, is it phosphorylates things in the alkaline pH of your intestine. So it must be down further in the GI tract. Okay, but it's waving. So this would be the surface of an intestinal cell, and this would be the lumen of your GI tract, and then various things that have phosphate groups come along, like what would be something that you've eaten that has phosphate groups? It's like any animal kind of protein is going to have DNA that's been sliced into phosphonucleotides, and so you want to get rid of those phosphate groups before you transport them across the intestine. So alkaline phosphatase can whack that phosphate group away from the nucleotide. Yes?
Is there a reason why it's anchored twice?
It's what?
It's anchored twice to the leaflet instead of
Oh,
just
it's
one?
because it's used. It has to be because that's the way fatty acids are set up for the phospholipids. They're phosphoglycerides is the correct term for a fatty acid anchored, classic membrane lipid. Okay, but it's that's the structure of any phosphoglyceride lipid. But you see. That one has two, but so do those other ones I talked about. So it's kind of like a rule, isn't it? Now I think we could expect that if you're having to hold a protein into a membrane as an integral protein, you're going to need two anchors, and this just naturally has two fatty acids, just like C-Sarc and just like RAS on this side of the membrane. Correct? Everybody's got two lipid anchors. Does that make sense? Yeah. Okay, and so this one does too. Now, another good example is alkaline phosphatase in your GI tract. And then, of course, there are certain diseases that are caused by protozoans. And one of them is called African sleeping sickness. And it is a really tricky pathogen. So the protozoan is Trypanosome brucei. Some brucei. Okay, and so this little guy gets in your bloodstream and does nasty things and causes African sleeping sickness. But what's so hard about it is it continually fools your immune system. So what it does is it displays a protein called a VSG. That's variable surface glycoprotein, and that variable surface glycoprotein is a GPI anchored protein. So it's cloaking itself, okay, with this little protein, and there's one over here. And so basically, it's using it as a cloaking device, right? Just like the Romulans. Okay, except this is real, right? That's in the 51st century or something. This is now, okay? And so what happens is, you make an antibody to this, your immune system tries to attack, you know what it does? The little kid lets go of the balloon. It sheds the GPI anchored system, and it makes a new protein that the immune system doesn't recognize. So it's cloaked. The immune system will not recognize it. So you have to attack it in different ways. So you can do all kinds of weird things, but it's also be put to use by pathogens. All right, so those are three good examples of GPI anchored proteins. And remember, they tend to have a high affinity for lipid rafts. So that's worth remembering. Okay. Now we go, what's the next step? Now we're into the peripheral category. And these are things where you can take the cells, and if you treat them with like high salt or extremes of pH, high pH, or if you take all the calcium out, the peripheral proteins will come out of solution. They will come and leave the membrane. Okay? So that's how we categorize something as a peripheral. But they actually are peripheral to the membrane. So instead of being anchored into it, they are just touching it in some way. There's two ways for a peripheral protein to be connected to the membrane. One is direct and the other is indirect. So what's a direct peripheral membrane protein? This one's a good example. So these are these little, they were supposed to be lollipops. So these are the phosphoglyceride lipids, okay? And remember that surface is bumpy, isn't it? It's something that a protein can recognize. And this protein called annexin will directly bind to that surface. Now, does it attach to it pretty weakly? Yeah. Those are all non-covalent bonds that it's grabbing onto. It's not forming a covalent link to the phosphatidylserine. It's just sticking to it with hydrogen bonds. And so if you put high salt in there, what you're going to do is disrupt the hydrogen bonds, and annexin will come floating into the cytoplasm. And then you can break up the cell and you can isolate annexin. Okay, so why did I choose this as the poster child for direct? Okay, annexin has a very special relationship to phosphatidylserines, and one of the things that we can say is if you're a living cell, most of your phosphatidylserines are facing the cytoplasm. But when a cell decides to kill itself, undergoes apoptosis, okay, cell death, cell suicide, what it does, one of the first things it does is it takes the phosphatidylserines and flips them to the outside. It's displaying those to the local lymphocyte saying, I'm ready to go. It says, I'm killing myself, so you might want to clean me up. Right? So a cell that's undergoing apoptosis can be recognized by the fact that it's displaying serines. But we can't see those. Right? So what you can do is you can take annexin and you can label it with a fluorescent molecule, and we can put those cells in culture, and we can actually identify the ones that are killing themselves if they bind to fluorescently labeled annexin from the outside. So you make annexin that's not in the cytoplasm, you bought it as a reagent, you put it into cell culture. If this cell type is about to kill itself, it will display the phosphatidylserines to the outside, and annexin will bind to it and light it up like a firecracker. Okay? So it's a good way of identifying cells undergoing apoptosis. Good. Okay. And then there's lots of examples of the second type of peripheral protein. It's called indirect. So an indirect peripheral protein is different because the way it attaches to the membrane is it binds to an integral membrane protein. So it's a Klingon. Lots of Star Trek stuff here. Romulans, Klingons. All right, so this is the Klingon protein because it's not directly touching the membrane, so it needs to have a binding partner that's integral. Okay. And so there's all kinds of examples of that. Let's see, I think I have one there. Here we go. So this is the red blood cell membrane. And red blood cells have to crawl through really tight spaces, capillaries. And so sometimes it's one cell, single file, going through a capillary. So it needs to be able to bend but not break. So because the plasma membrane is kind of floppy, it's okay. It'll squeeze through a capillary by itself. But the inside of the membrane needs to not bust. Okay? And what keeps it from breaking or bursting is the cytoskeleton is lining the membrane and giving it a skeletal support. So this is a protein skeleton scaffolding. And the way this scaffolding is constructed is take a membrane protein. So BAN3 is a transport protein. It's actually a bicarbonate chloride antiport. And we're going to talk about that in a couple of lectures. Okay? Antiports. So it takes a chloride, and in exchange for moving it in, it brings a bicarbonate molecule out. So it's an exchanger. And one of the rules that you'll find out very quickly is transport proteins are almost exclusively multi-pass membrane proteins. And that's true for BAN3. Now, why is it called BAN3? Why don't we just call it an antiport? Well, it was isolated a long time ago. What they did is they solubilized the membrane, and then they used a technique that we'll talk about and that you use in Cell Lab called SDS Page Gel electrophoresis. And they ran all these bands, and each band is a different kind of protein. And it was band number 3. So that's how it got its name. Band 3 is a transporter. And it is the integral membrane protein that is the hand grip for this protein here. That little magic ball, that's not a globe, that's a protein called ankyrin. It tells you exactly what it does. It's a peripheral membrane protein that's anchoring the cytoskeleton onto band 3 in the plasma membrane. So the linkage is band 3, ankyrin, and then you have these little curlicues. You saw that in the video, actually, in day 1. This showed up, right? And it was the underside of the plasma membrane, and this molecule is spectrin. Okay, I think that's all in the notes too. Okay, so the key thing is you can use peripheral membrane proteins either directly binding to the cytoplasmic leaflet, or they can hitch a ride and be a Klingon and be an indirect peripheral membrane protein. And so ankyrin is a poster child for that group. All right, questions about integral, membranes, peripheral. So you can get a pretty good concept map now. So you have membrane proteins at the top, and this is a good exercise for you to do. And then break it into peripheral and integral, and then keep breaking it down into lipid anchored on the peripheral, on the integral side, and multi-pass, single pass, and then you break it down and then give me examples of each one. That's a pretty good concept map that will make you remember this stuff. But any questions before we leave the vocabulary? Yeah.
Did you say ankyrin was a poster child?
For the indirect membrane protein. Indirect peripheral membrane protein. I mean, you could, if you don't like that one, you could use band 4.1. What's its binding partner? Glycophorin. And band 4.1, and so you have this nasty mess. That's why I'm staying away from it, because it's more complicated. But it does the same thing. Okay. Could you use spectrin as an example? Absolutely. All of this is attached as indirect membrane protein. So everything that's not an integral membrane protein that's stuck on the membrane is a Klingon, right? So spectrin is a peripheral membrane protein, indirect. So there's lots of examples. But the one that you need to know is anchored. Okay. I think we're talking about, let's do this. One of the, I've already mentioned the heterogeneity of the different leaflets, but sugar groups are usually on the exoplasmic domain along with disulfide bridges. I brought that up. But it's good to remember that rule, because it will help you tell which side of the protein is facing which direction, if you know where the sugars are attached or where the disulfide bridges go. All right, so what we want to do is get into this. We're going to construct a membrane toolkit that we can use to look at some of these proteins. And one of the techniques that we use is fairly infrequently is called freeze fracture electron microscopy. It's standard transmission electron microscopy. And basically, in a nutshell, you take the sample and you get it really cold, and then you crack it with a diamond knife or some device. And what happens is when you fracture cells, the fracture lines tend to go between leaflets. And then you can peel them apart and you can coat them with metals, because when you're beaming it with an electron ray, cell components are not electron dense. The electrons will just go straight through. So what you do is you make a replica, and again, you don't need to know the technique to that detail, but you make actually a metal shadowed replica, and then you can actually get these, you can tell the distribution of certain proteins. So for example, this is a bacteria that expresses bacterial rhodopsin. And so this is a patch that this bacteria is using to use light as a proton pump. So it's receiving light, and then in exchange for that light, it's using the energy of sunlight to pump out protons. So this is the battery in that bacteria. And the way it does is, like a photo array that you use to turn sunlight into electricity. This is its photo array, except it's an array of bacterial rhodopsin, those 7 multipass membrane proteins. They're coalesced into a little patch that's designed just to collect sunlight and pump out protons. And we can tell the distribution or the aggregation state of different proteins using freeze fracture. So not used a lot, but it tells you whether they're all scattered around or they aggregate into a particular place. So I'll give you an example of where we use that later on when we run upon it. But we'll put that in our toolkit, right? Okay, so then this is SDS Page gel electrophoresis. And what's happening is if you cast the gel and you make it out of something called polyacrylamide, what happens is you polymerize that gel, and then we're going to force proteins to move through the gel. And depending on what concentration of polyacrylamide that we use, we get gel pore sizes of different sizes. And so we're forcing the proteins to go through the gel by pushing them with an electric charge. And so that's why you put an electric field across this gel. And so the proteins are going to run so the proteins we actually coat with a detergent called SDS, sodium dodecyl sulfate. And it coats every protein with the same type of charge. They're all negatively charged. And the reason we do this is there's three properties that is going to control the movement of that protein through the gel, and we want to cut out all of those properties except one, molecular weight. So how do we do that? So you get your gel, and it helps to know a little background chemistry. So there's three things that are going to, so let's put our gel over here, and we're going to load our protein as a clump right there, and then we're going to put electric field, and we want to put the positive at the very bottom because all the proteins are going to run to the positive because they're negatively charged. So we're going to put them at the top, and so as soon as we turn on the electric field, it's going to start to migrate through each lane of the gel, and it's going to go through polyacrylamide. Okay, there's three things that drive its movement, and the first one is the charge to mass ratio. Okay. Now, typically, if you didn't coat proteins with SDS, they all have different charge to mass ratios. They're different sizes. Some of them have lots of negative charges. Some of them have positive charges. It's a mishmash. So we don't want that to be a variable. So the way we'll get rid of that is we'll take SDS, which is a negatively charged detergent, and we're going to take that protein and we're going to coat it uniformly with negative charges. That's where the sodium dodecyl sulfate detergent comes in. Okay, so it's going to get rid of that ratio. Every protein is going to have the same charge to mass ratio, so that will not determine the migration rate through the gel. Okay? The second thing that determines the migration rate through there is the molecular weight. Okay? We want that to be there because that's how we're going to segregate the proteins into bands. Band 3, band 10, whatever, is by molecular weight. And what does the choosing of where it goes in the gel is the gel size, the pore size in the acrylamide. So for example, if you give a 5% level of polyacrylamide in the gel, that means it has big gaping holes in the gel. And the only kind of protein you can tell apart is big proteins. So if you want to tell big proteins apart, the only kind of gel that will, the type of gel that will best resolve the big proteins has big pores or low percentages of polyacrylamide. So a 5% gel, there's a figure here, will distinguish proteins. So if you use a 5% gel, it will resolve 60 to 250 kilodaltons. Okay? What if your protein is small? What if it's a peptide? What if it's short? So if it's below that, what are you going to do? Take a guess. So it's pore size you're after. You want smaller pores. What do? You increase the percentage of polyacrylamide. So you can make let's see, what's a good example? A 15% gel. And then you can resolve proteins between 10 and 50 kilodaltons. So the higher the percentage of acrylamide, the smaller and more selectively pores you're creating that you're forcing the proteins to move through. Okay, so we want that property to be the same. We want to keep that property, and so we're not going to eliminate that one. But the third thing we have to get rid of here is molecular shape. So clearly, if you're shaped like a snake, you're going to be able to wiggle through that gel faster than if you're a big, bulky ball. So how do we deal with that? We denature everything and turn everything into a primary sequence. Everything is turned into a snake. So that's called denaturing. So we use a denaturing gel. What does that? SDS. Because that negative anionic detergent will actually coat the protein and break up all the ionic bonds, all the polar bonds, all the hydrogen bonds, and it will just come out like a snake. Completely unwind it, and it's uniformly charged now, and everything is a snake. Okay? So everything has the same shape. So we've eliminated the charge to mass ratio differences. We've eliminated everything is a snake now. The only thing we've left is the only thing that sorts the protein. So they're all sorted on the basis of molecular weight. So when these things run on the gel, there's a different color. So if you're a blue protein and you're up here, when the gel stops running, and you're a green protein, you're down here, what happens is this is the higher molecular weight. So the ones higher in the gel that migrated shorter distances, they couldn't get through the pores, right? It took them longer. But the little guys snake through here very quickly. So this is decreasing molecular weight. And how do you know where 50 kilodaltons is versus 10? Well, you put a molecular weight ladder of standards. You know the molecular weights of all these proteins, and then you can compare where your protein's weight is compared to the molecular. It's called the molecular weight ladder. So we can resolve from this the molecular size of that protein. Yes?
Is it a beta mercaptoethanol that linearizes the protein?
Yeah, so the other side of this is if your protein and membrane proteins that are on the display, this is a good test question. You're giving that away. No, it's okay. If you're, like you have a multipass membrane protein and part of the protein faces the outside, what's it going to likely? It's going to have two things that are likely. Sugars and disulfide bridges. So you're going to need to break, the SDS will not denature the disulfide bridges. So you put beta mercaptoethanol. Let me write that down. Beta mercaptoethanol. And what that does is it reduces the disulfide bridges, and that really unfolds it. Okay. This is just wonderful to work with. This is what the gas company puts into natural gas to give it a smell. You do not want to spill this on yourself, or people will not talk to you, particularly on Valentine's. Do not give that to your special other person and have them sniff it. Yes.
Is that why they use SDS page rather than urea?
Yeah, because it's not going to leave a uniform charge.
Oh.
See, urea would do it. Now, you can get the peripheral proteins off with urea. That's a good thing. So if you're just interested in peripheral proteins, you can do that and then dialyze all the urea out and then put the SDS, blah, blah, blah. So you could use urea for certain steps and not others. Now, but what if you want to identify the proteins? We can turn this into a Western blot, right? We don't do this in our cell lab, but in the experimental cell lab that's now being offered, they take this one step further. They actually blot these proteins onto paper, and then you can use antibodies that are labeled to light up specific proteins. So you can tell exactly which protein is yours, and the bigger the amount of protein, the bigger the band, right? Because it's related to how much is there. So there's a lot of things you can do with this technique, but this is the starting point. Okay, what do you do with those sugars? Uh-oh. Will it run differently with glucoses all over it? Or sialic acid is the worst one? Absolutely. It messes up the charge. So what do? You put a non-specific glycosidase in there, and you chop off all of the sugar residues. Glycosidases. to get rid of the sugars. And now you've gotten rid of the sugars, you've gotten rid of the disulfide bridges, you've turned them into snakes, and you've coated them with negative ions. The only thing it's going to run is based on its molecular weight and how fast it can snake through the pores that you determine. You're in charge of everything. Okay. That's enough techniques there. Any questions with that before we move on? It's pretty straightforward. But you can see how your knowledge of protein chemistry is really going to help you. And there's a picture of that sulfur group is white, it's got a negative charge, and there's beta-mercaptoethanol. I'd forgotten that I know that. So, yeah, the key thing is that SDS has another function. Because it's a detergent, that means it's amphipathic. It's got a charge and then a long, where's it got? See, it's got that long, this is the fatty portion. That's what a detergent does. It will actually break up the membrane. So that protein is not coming out of there unless you use a detergent. You pull it out, and then you actually replace all these lipid molecules with SDS. Good. Otherwise, it won't come out. And there's all that. Okay. We'll just barely get into this, but the last technique I'll talk about today is what if you want to try to identify a protein that potentially has alpha helices, has transmembrane alpha helices. Is there any way to do that without actually completely isolating the protein? Now here's a case where you would want to do this. You've got the human genome has been completely sequenced, and so you have the sequence here, and somebody asked you the question, are there transmembrane alpha helices in this protein? Therefore, is this a membrane protein is really what you're asking. And so you don't have to go to the trouble to make the protein, crystallize it, blah, blah, blah. You can actually scan it with something, and you can use what's called a hydropathy plot without ever touching the protein. As long as you know the sequence, you can get an idea of whether it might contain membrane-spanning alpha helices. Okay, so let's understand what we're looking for. The property that you're looking for is something that has about 19 to 23 amino acids long, and that span of amino acids must have what property? Look at it. They're all family BWR, PG, or whatever that thing was. They're all hydrophobic, right? So there must be a window of amino acids that are negatively charged. I'm sorry, that are hydrophobic. And so what we do is use this plot, which is called a hydropathy index, and we plot it versus the amino acid sequence on the x-axis. So what you do is you get your sequence of amino acids, and this is the N-terminus, always the N-terminus. That's the C-terminus. So this is how long that protein is. And the sequence is there. You know which amino acids because you have the gene structure, correct? Okay. Each amino acid has a hydropathy index to it. And how do you get that? What they do is the hydropathy index is the measurement of the free energy change of taking that amino acid from oil and moving it into water. So if you have let's do an easy one. So if you have a hydrophilic amino acid, give me an example. Hydrophilic amino acid.
Lysine.
Pardon me?
Lysine.
Lysine, positively charged, very hydrophilic. Okay. And you put that in oil, is that amino acid very happy? No. Okay. So if you move it, it's got a high free energy in oil. And if you move it into the aqueous phase where it wants to be, what's going to happen to the free energy? It's going to decrease. It's going to be so what that means is negative free energy changes are associated with polar and charged amino acids. So the more negative it is, the more polar that amino acid is. And all those show up down here underneath the zero. So if it's kind of neutral, if it doesn't know whether it's hydrophobic or hydrophilic, it's sort of sorting around in here, right? Now, if you're hydrophobic and you put it in oil, it's extremely happy, right? It's got low free energy. What happens when you move it into the water phase? Actually, it'll stick to each other, right? And so it has a positive free energy. It's very unfavorable. That movement is very unfavorable. So all residues that are above this line have a positive free energy hydropathy index. So plus values are above and hydrophobic, negative values or below the 0 and negative. And so we can get a value for each single amino acid. But we don't do it that way. What we do is you have to move out here to about amino acid number 11. And then you take the 10 amino acids on this side and the 10 amino acids on that side. That constructs the hydropathy window. And you average all of those on both sides of the one you're interested in. And you come up with a net sum of the hydropathy indexes. And that puts you on this graph. If it's negative, you're down here. If it's positive, you're up there. Okay? Now, what we're looking for is we're looking for peaks in the hydropathy index that have the right number of, they need to have the right number of amino acids, 19 to 23, and they need to have a peak in here because these are all going to be pretty much hydrophobic. Not exclusively. If you notice, there's some violators in here. You know, they didn't get the message, right? So let's find one. Histidine, now that's pretty much on the surface. Glycine, cysteine, tyrosine. See, there's tyrosine. It's very happy in there. It's actually pretty close to being polar, but you go down here. Well, this one's a little weird. See, histidine, that's still in the membrane, but it might be close enough where it's taking a peek at the water, right? So it's happy. So anyway, it has to have the right sequence, and it needs to have this peak in hydropathy. Right? So if you're right there, it means 10 of your buddies on the left are hydrophobic, and 10 of your brothers on the right are hydrophobic, and that's the middle of the membrane, the very middle. It's about 20 amino acids, so you're amino acid number 10. Right? Okay, so certain distance, and it needs to have a certain hydropathy index, which would be positive. And so what you can find, so this is glycophorin, as you saw, it's a single pass. And so all this stuff is nice and polar. And let's go back really quickly in the remaining minute. Let's see if we can go back to the picture of glycophorin. Well, we had it there. I skipped past it. Go on. Yeah, there we go. So one of these, I can't remember if this is the N-terminus or that's the N-terminus. But you can situate this in the membrane based on where that alpha helix is. So this is either cytoplasm, and there's actually a way you can figure that out. But what would the one Bacteriorhodopsin look like? How many peaks would it have? 7. And you're right. See, it gets a little messy here, so it's not as clean as you might like it, but it does fit the bill. Okay. We'll continue this next time.